pandemic SARS and pandemic influenza. Kanta, thank you.
Well, thank you very much. And I'd like to thank the organizers for this opportunity to speak to you today. Many people in this room have worked on influenza for several years, including myself. And of course, we've been in the midst of the COVID-19 pandemic for the last 3 years. So this is a slide that summarizes the latest data from the WHO on the numbers of SARS-CoV-2 cases. And if you can read the fine print, it's basically we're up to 750 million cases. So, it's an incredible number of cases over the years. However, the great success has been the rollout of vaccines, and that has certainly changed around the world. But there are many features that are shared between pandemic influenza and pandemic coronaviruses. And I'm going to talk you through some of these. Both emerged from animal sources. There's evidence of, there's the ability in both sets of viruses, both families of viruses can exchange genetic information. Airborne spread that has come up already a couple of times. We'll talk a little bit more about that. Genetic and antigenic drift. Our great hope at the beginning of COVID-19 was that it would be like measles. We'd have a vaccine that would last us 50 years. But in reality, we're living with a lot of genetic and antigenic drift. And so I'm going to close by talking about what the implications of that are for vaccine strain composition, which is a very familiar theme for influenza virologists. So this is a diagram that sort of shows you how coronaviruses occur in nature. So we've got viruses that almost every species has their own coronaviruses. And typically, coronaviruses stayed within their species. There was little crossover between species. There were some examples, but largely they stayed within their species. So here, this is a review from Rachel Graham and Ralph Barrick, showing you the bat coronaviruses were blue, human coronaviruses were green. There were domestic animals that include cows and other domestic animals, red, and they rarely really crossed over. But what we have with a changing ecology, loss of habitat, sort of crowding and in urban settings, and lots of travel, we are encroaching on each other's worlds. And all of these viruses have the ability to recombine their genomes. And so we see more and more evidence of recombination and crossing species barriers. On the other hand, influenza viruses don't recombine very frequently, although that was the textbook claim that they don't recombine. There are isolated instances where they do, but largely influenza viruses undergo major genetic change by what we call reassortment. And this is a consequence of the fact that the genome is comprised of multiple gene segments. So influenza viruses have 8 gene segments shown in a schematic. In a blue virus has 8 gene segments. The red virus might be a human influenza virus. If they infect the same cell, each of the gene segments is replicated. And when new daughter virions form, 8 gene segments typically from each one come together to form an infectious daughter virus. But if there's a soup of gene segments that have been replicated in that infected cell, you can have a progeny virus that derives 1 or more gene segments from the other parent. And that is in fact how reassortment occurs. We know that the pandemics of 1957 and 1968 were caused by avian-human reassortments. And we know that the pandemic of 2009 was also caused by a reassortment virus where the reassortment occurred within animal species and then crossed over into humans. So we do know that there's a lot of reassortment. We now recognize that there's a lot of reassortment that's happening on an ongoing basis, even amongst seasonal influenza viruses, and certainly in animal influenza viruses. So there are 2 different mechanisms by which these families of viruses exchange genetic material, but both have the capacity to significantly change their properties. So the major modes of transmission of respiratory viruses are sort of outlined on this schematic. And it's a lovely, very thorough review by Nancy Lung in Nature Reviews Microbiology, and truly amazing because it's a single author review. It's exhaustive. So what she's pointed out here is that you've got the possibility of infection from fomites, where virus lands on a surface and then you might infect yourself by direct contact, which is shown by that hand-to-hand contact, but also respiratory transmission, which could be either from larger size droplets or finer droplets. The finer droplets persist in the environment, in the air for longer. So this sort of summarizes the various mechanisms. And one of the things that's come out during the last 3 years of studying COVID-19 is the recognition that the biomedical literature uses very different terms to describe airborne transmission of pathogens than the aerosol scientists do. And so here is a summary from a US National Academies workshop in 2020 that brought together people from aerosol sciences with people from the biomedical sciences and infection control and infectious diseases. And we, at least I in medicine, I was taught the magic number was 5 microns, less than 5 microns, it stayed up in the air and transmitted long distances. More than 5 microns, it fell out of the air within 3 feet or 1 meter, depending on where you live and how you measure distance. And that was why beds in hospitals were 1 meter apart. Turns out the aerosol scientists say absolutely not true. There is this 5 microns is complete nonsense. And that in fact, their definition of particles is that they could be as big as 100 microns in size and they rapidly evaporate. And so there's a real desire to bring the two fields together to inform each other and change from that traditional thinking to these newer definitions. And that really talk about the aerosols versus droplets and how they can transmit, and importantly, how they can be managed or mitigated. So there are a number of factors that affect airborne transmission, and infected host produces, everybody produces aerosols. So as I'm speaking, I'm producing aerosols, but whether they contain virus depends on whether I'm infected or not. And there are particle characteristics, there's distance that matters, there are environmental conditions that matter, and of course, the host. So several things affect airborne transmission. And some of the key points that we've learned from SARS-CoV-2 and air research is that droplet transmission dominates when people are within 0.2 meters of each other, if they're just talking, and 0.5 meters of each other if there's coughing involved. Viruses are enriched in aerosols, but there's a lot of interpersonal variability. So think back to hearing about the super spreader event. So that 20% of cases were actually responsible for 80% of the infections. Aerosols are the main mode of long-range transmission, but aerosols also play a role at short range. So that whole idea that 5 microns, less than 5 microns wasn't an issue if you're close, you're still in what people refer to as a plume around a person. And only aerosols, not droplets and not fomites, are affected by ventilation. Transmission is favored at low humidity. And of course, as I pointed out, they're super spreading events that we don't fully understand because they tend to be defined in retrospect. So the key point that I would say is that airborne transmission of influenza and other respiratory viruses should now be reassessed with these new concepts in mind. So what about vaccines? The vaccines for SARS coronavirus 2 were developed based on principles that had been established for SARS-1 and for MERS. And so we already knew that the spike protein was the main target of the protective immune response. We knew that you could present that spike protein and induce neutralizing antibodies, and that neutralizing antibodies would confer protection. And there were many people don't know, but there were lots of vaccines that were developed for SARS-1. Only 2 went into phase 1 clinical trial, but there was a tremendous amount of work that had been done. So science is an iterative process. And so we learned from the principles established with SARS-1 and MERS, moved on very quickly to SARS-2 in the development of spike-based vaccines. And this slide demonstrates the number of platforms that are available. Some platforms have been used in certain countries and others in others, so it's really not a uniform use. We certainly hear a lot about the mRNA and viral vector vaccines because they were in use in Australia. Protein vaccines have been introduced. We haven't used inactivated vaccines or DNA vaccines, but they've been used in other countries. So as I said, we were very hopeful that we wouldn't have to update these vaccines and that would be one vaccine that would last us through to control this pandemic. But unfortunately, as we all know, a number of variants started emerging. And you've heard from previous talks about the alpha, beta, gamma that we didn't see, delta, and Omicron. And these are slides, these are snapshots from NextStrain. If you haven't visited this website, it is fantastic. And they have a lot of influenza data and a lot of COVID-19 data. And on the top graph, you're looking at the whole period from the beginning of the pandemic to the present time, showing you the emergence in these colored clouds of the various variants. The next slide down shows you the same data, but now the lineages are pangolineages. So the terminology is a bit different in the next slide. And then showing you, contrasting what's happening on NextStrain, you can toggle between different regions of the world. So showing you what's happening in Oceania versus North America. And so in North America right now, it's the XBB and XBB 1.5 that they're concerned about, whereas we've got XBF. So there are differences in different parts of the world. And that becomes important when we consider what the implications of this are for vaccine strain composition. So you've heard already that, so the vaccine is based on the spike protein. The spike has significant sort of functionally important parts. There's the NTD, which is the N-terminal domain, the RBD, which is the receptor binding domain, within which there's a receptor binding motif, which is the RBM shown in turquoise. And then there's the S2 portion of the molecule that the entire spike is a very, very large protein that then has to be cleaved. So what I've got on this slide are some of the key mutations that have been seen in alpha, beta, gamma, delta, and Omicron BA1 and BA2 variants in the RBD and in the receptor binding motif. So you might, if you, and I suspect you can't see the details from far away, but you will note if you did have the time to look at these numbers, a lot of these residues actually are the same residue where they are toggling back and forth and changing. We see this in flu. We're used to seeing this in flu. But so at this point, I think we are gradually reaching a point where it is less important to call it by a Greek lettering and more to recognize that if we see a change at amino acid 484 in the spike, we need to worry, or 452. So there's certain hotspots where the virus is actually experimenting and changing in these residues, either driven by immune pressure or to achieve better binding to the receptor. Again, this is now showing you beyond BA1 and BA2 that just huge numbers of mutations that are occurring in the spike protein. So what do we do about this? And how, if any, how do we take this into consideration in terms of vaccines? So what the WHO has done is they have a series of technical advisory groups. The first one is what focuses on the virus, on monitoring the evolution of the virus. And that's, so the technical advisory group of abbreviation is TAG. So TAG-VE is the technical advisory group on virus evolution. So they monitor the changes that are occurring in the sequences that are deposited into GISAID or elsewhere. And they monitor the effects of those mutations on transmissibility, immune evasion, receptor binding, and a number of other properties. The second group, there are two groups that now look at the vaccine and the implication for vaccines. One is an R&D blueprint that many of you would have been on calls with. They organize webinars and advice discussions And then there's a tag COVAC, which is what I'm going to talk to you more about, which has been developed essentially as an advisory group to advise on vaccine strain composition. And then at the policy end is SAGE, that advises on vaccine use and implementation. So tag COVAC is an advisory group to the WHO that was put together to make recommendations on the methods to assess the impact of the variants of concern on vaccines, to provide an interpretation of the available evidence on the effect of those variants of concern on vaccines and vaccine effectiveness if possible, and importantly, to recommend to the WHO for each COVID-19 vaccine platform whether adaptations are needed in the vaccine strain composition so that they continue to provide the appropriate levels of protection. So this is what the committee looked like a year ago. I chair the committee. I said I would do it for a year, and I'm going to do it for another year. I don't know why. And David Wentworth, who's also card-carrying influenza virologist, but also a card-carrying coronavirologist, is the deputy chair of the committee. We've got 18 members, and there's been a little change in that role for the second year. And Rhina McIntyre from Sydney is on that group as well. So the things we considered when we first started this discussion was whether, and we started talking about this when it was Delta. So that's how long ago we started discussing vaccine. And we asked whether there was an update that was needed in the composition of the vaccines, and if so, what would the threshold be to do so? And we talked about the approaches, and the world is divided into people that think that influenza virologists know what they're doing, and there's a group that thinks that we don't. And so there were many people who said, well, influenza is the only vaccine that is updated every year. Let's see how they do it and learn from that. And there were other people who said, why should we do that? They don't do so well. However, so we had the options of building something completely de novo or trying to build and make it relevant and better. So the mechanism is this tag COVAC. We interface with regulators, and we are attempting to interact more and more with vaccine developers. And we certainly interact regularly with the tag VE and to some extent with SAGE. So what we decided early on was that, as I said, since influenza vaccines are the only vaccines that are updated on a regular basis, that we should look at the type of information that we use to make influenza vaccine strain composition updates. And this includes virologic and disease surveillance data, genetic characterization data, antigenic characterization, including antigenic cartography, human serology data, where we get post-vaccination sera from different age groups and different geographic locations, and see how those sera react with new variants, predictive modeling, and vaccine effectiveness data. So we have now attempted to get as much of this information as possible for COVID-19 and to utilize it for these decisions. So the last statement that we published was in June of last year. So it was a recommendation about Omicron, at which point BA1 and BA2 were circulating, and BA5 was just beginning to emerge. So we used the information on the evolution and spread of SARS CoV-2, the available data on vaccine effectiveness of the ancestral vaccine against Omicron strains, some cross-neutralization and cross-protection data, antigenic cartography. We had some preliminary information on Omicron infection and some preliminary data on candidate vaccines with updated compositions. And at the time we had these discussions, Omicron was the most antigenically distinct of the variants to have emerged. And at that point, BA1 was as far away from as any from the index virus. Antibody responses in previously unprimed, so previously naive individuals who had been exposed to Omicron had strong responses, but not great breadth. So if you had a naive individual who had a primary infection with Omicron, they made a great response to Omicron, but not so much to the other variants. However, if somebody had been previously infected or previously vaccinated and then had an Omicron infection, they not only responded well to Omicron, but they also had great breadth. And so these data supported a preference for the inclusion of Omicron in an updated vaccine, administered as a booster dose. So this is a picture of the antigenic cartography data at the time, showing you the cluster of viruses in the top left, which are all the original virus and the early variants, and Omicron BA1 and BA2 as far away as possible. That's the red dot and the, looks to me, kind of brownish dot. We did not know what the trajectory of Omicron evolution would be. We didn't know if the next variants would continue to come off the Omicron lineage or would flip back and come off Delta. We didn't know that. We also knew that the original ancestral vaccine gave very robust protection against severe illness, hospitalization, and death, and in fact continue to do so. And we didn't know, because of the limited amount of information we had with primary Omicron infection and primary Omicron vaccination, we didn't know how good the breadth would be of protection should a variant come off the original. So given all of these uncertainties, we did recommend that the original ancestral strain be continued in the vaccine and the option to include an Omicron strain be included. I want to make one very important point, and that is that in this discussion about SARS-CoV-2, we really want to emphasize that we're not chasing variants, that what we're trying to achieve is improving the breadth of immunity. And so we really don't want to be discussing whether we're always going to be behind. We really want to change the concept to breadth of immunity. So we did not advise dropping the ancestral strain. We didn't advise using a monovalent Omicron strain at the time, but now we have quite a bit more information. And so at this point, what was done in 2022 and what the future will hold are that the mRNA vaccines were updated. A bivalent ancestral plus BA1 vaccine was used in the UK and in Australia and in Europe. A bivalent BA5 vaccine was used in the United States and is now licensed in Europe. Our committee is planning to review the data from the use of these updated vaccines and the use of a monovalent ancestral booster to see, did it make a difference? Did just a booster matter, or did it matter that the booster had Omicron in it? And if it mattered that Omicron was in it, did it matter whether it was BA1 or BA5? I think this is going to be very valuable in informing us how to make decisions going into the future. What do we have to do to achieve that breadth? So we're discussing the timing and frequency of strain composition meetings. The US FDA has already committed to an annual fall, winter revaccination campaign, and they're proposing a strain selection annually in May or June, but we're still in the discussion process because the US FDA decision is only about the United States. So what's going to happen for the rest of the world, including us, is something that the WHO is still discussing, but it looks like we might be doing 2 decisions a year, or at least 2 meetings a year to decide. So with that, I'm going to stop. I'm 3 minutes over, 3 minutes and 20 seconds over, but thank you very much for your attention.
Kanta, thank you. Plenty of time for questions. Bill.
The bivalent vaccine with ancestral being BA2?
No, ancestral is Wuhan.
1. And is...
Not BA1, Wuhan.
Okay.
Yeah.
So is the logic that you'll broaden the immune response by giving both, or is the logic that you've got a similar prime boost as you have with the BA5 post-vaccination or post-infection, or is there...
So I think the best way to describe what we were doing a year ago was hedging our bets. And so we knew a lot about the Wuhan-based vaccines. We knew a lot about their performance. And we were not comfortable dropping that completely and going to Omicron alone. I think now we're going to be looking for data on Omicron monovalents in addition to Omicron-containing bivalent vaccines to understand that.
Alan?
Yeah, my question's about the sort of global governance of all of this. So immediately after you put out that, statement and explained it to VRBPAC and us at ATAGI and so on, the FDA just said, oh, we want the BA5, we don't care about BA1 anymore. is that likely to be an issue going forwards, or do you think now there's a bit of sensibility returning to the?
I couldn't comment.
Very
I think we probably do is have our meeting before VRBPAC. But we did even before. So there were a couple of things that surprised me, quite honestly, about the FDA decision, because I think it was quite a departure from their usual to say they would not require human data. And the reason we recommended BA.1 was that's the only strain for which we had human data. So we were very surprised that they were willing to go out and say that they would allow animal-only data.
Terry?
Kanta, thanks. Fantastic talk. For recombinant protein vaccines and thinking of flu, for example, looking for breadth is where you look for adjuvants to give you breadth. And with the increasing precision of targeted, prescribed targeting with mRNA in particular, we're actually shooting ourselves in the foot by becoming maybe too precise about what the targeting is of the epitopes. What do you think about the adjuvants, the adjuvants in the, for example, the Novavax adjuvant or the other adjuvants used in flu vaccines, both first generation and latter generation adjuvants potentially to be now, we need to be thinking about that potentially for SARS vaccines.
Yeah, I think it's a complicated scenario, because I think that there are a couple of ways that you can achieve breadth. One is just raise the magnitude sufficiently against all variants that you get there, or you use an adjuvant to give you breadth as well. So I think there might be many ways to get there. And I suspect actually that a combination of platforms might be ideal. The problem is that we are not in a situation where we have studies where we're able to compare these different platforms with each other. Even the assays that are being done are not identical. So the Novavax assay is not the same as the assays that we're used to seeing with the mRNA vaccines. So I think that is an additional wrinkle that I think the regulators really struggle with.
Coming back to air quality before, just to take it a bit away from the vaccines, Kanta, what's the issue then about having air quality control? Because it seems like there is not any discussion in Australia or elsewhere about how we can actually minimise transmission across.
I think there absolutely must be. The science is there. The ability, as Bill said, to recover infectious virus from air has really, I mean, the technology's improved dramatically. We have a couple of different instruments that we can recover infectious virus from the air. We can recover influenza. We've got a whole series of experiments in vitro showing how, what level of detection we can get. And we have a clinical study planned to use those instruments in SARS CoV-2. So I think that there's no excuse not to start having that conversation. And Australia has some very highly qualified aerosol scientists.
Just coming back to the earlier discussion, we've had a lot of discussion at previous IC meetings about antigenic sin. And so when you keep using the original variant, you run the risk of actually not boosting the strains that are around. Now, you presented data that that's not necessarily effective, but thinking outside the box, what can we learn from our understanding of antigenic sin with influenza vaccine that might inform us about better COVID vaccines?
So people have worried about this a lot. So the question is, if you keep the ancestral strain in a bivalent preparation and you keep vaccinating with it, that you will boost pre-existing memory B cells that were directed against the original vaccine, and you won't make new responses. That does not appear to be a problem. We are making new responses even with the bivalent vaccines. So I think I think it's not straightforward. It's divided still, but it's pretty new. So I think people are looking at that question, and it is complicated because in the bivalent vaccines, you used half as much of each of the strains. And so it is quite a difficult question to tease apart. But I think I haven't seen data that suggests that it's been detrimental to have the bivalent, the original Wuhan strain in the bivalent vaccine. Do we need it anymore? Maybe not. Do we have enough confidence going forward that an Omicron monovalent vaccine will give us the sort of breadth we need in a previously unprimed population, which admittedly is a smaller and smaller portion of the world? We don't know. So those are the data that we need to look for.
It seems to me that one of the hidden premises, and the FDA seems to believe that this is going to be an annual epidemic virus. It doesn't seem to me that that's at all established really. In fact, and the endemic coronaviruses are often biannual. So any thoughts on that?
Yeah, so I think it's too soon to tell. I think it's reasonable to have a committee. So I think that the seasonality of this virus, my own personal view is that the seasonality is not yet clear. The Beta and the Omicron BA1 emerged in very warm countries at very warm times of the year. So I'm not sold on the seasonality being winter, but people's behavior changes in the winter. So in temperate climates, I think there is a small blip in the number of cases because people stay indoors. But does that mean that there'll be no emergence? I don't think so. I think what's happening, what we have to worry about is that if you boost people and there is a bit of a decline in antibody at about 6 months, and it so happens that one of the variants that comes along with one of these 452, 484, et cetera, mutations, that could become a big surge. So I think the WHO is keen to keep an eye on things and have at least a biannual meeting to review the data. I think the US has said that they'll always have a backup of being able to call an emergency meeting, but I think that they're sort of with the wishful thinking of trying to make it more practical and easy and logistically palatable.
Kanta, fabulous.
Thank you.
And climate might change.
Yeah, that's true.
It's being suggested that we've got 2 minutes left, and so it's time to finish. I want to thank all the speakers for an extraordinary start to the resumption of face-to-face meetings. Long may they live. We might need to get air purifiers and stuff like that in the room, but we start again tomorrow at just before 9. So be here and with open minds and lots of questions. Thanks everyone for your participation.